Hello students, welcome to Biotech Academy. We are discussing Mendelian genetics, and today we'll talk about chromosomal theory of inheritance. Chromosomal theory of inheritance. This theory of inheritance, it identified chromosomes as the genetic material which is responsible for transmission of characters from one generation to the other, which was initially studied by Mendel, and who is also known as a father of genetics. So this theory, chromosomal theory of inheritance, it was studied by Walter Sutton, and this theory was studied by Sutton and Bovary. They worked out the chromosome. They worked out on the movement of chromosomes during meiosis. So they worked on chromosomal movement during a meiosis. So what happens that during the formation of gametes, during meiosis, which is a type of reductional cell division, the chromosome number reduces to half, and then after fertilization of gametes from opposite sexes, then it restores the actual number of pairs of chromosomes which are present at that particular species. So then this theory is used to explain the movement of the factors or the genes. The term factor was proposed by Mendel. So the movement of these chromosomes, it is similar to the movement of the factors which Mendel identified through his dihybrid and monohybrid and dihybrid crosses. And so this theory identified that these factors or these genes, they are located on chromosomes which follow similar pattern of movement from one generation to the other via formation of gametes and fertilization. So some of the important points or the important terminologies which we'll come across when we are going to discuss this theory. So some of the important terminologies are, the first one is autosomes. So what are actually autosomes? So autosomes are any chromosomes which are other than sex chromosomes. So there are the chromosomes, they are of two types. One is autosomes and the other is sex chromosomes. So sex chromosomes, they are the chromosomes which help in the identification of the sex, whether that particular organism is a male or a female, depending on the presence of sex organs and sex chromosomes. And other than, there are a number of other chromosomes which are not responsible for sex determination, and they are known as autosomes. So autosomes are other pairs of chromosomes, all other pairs of chromosomes which are different from these sex determining chromosomes. Then we have hemizygous. Then we have this term hemizygous. So hemizygous is having single copies of genes in an otherwise diploid organism. So the organism is actually diploid, but still then there are some chromosomes which are not present in pairs. So that condition is known as hemizygous state. And then the third term is wild type. Okay, so wild type is the typical form of an organism or a strain or a gene or character which occurs in nature. So that is known as a wild type. So these are some of the terms, and some of the important points about this chromosome theory is that these homologous chromosome pairs are the pairing, and their distribution from one generation to the other is independent of the other. So because suppose in case of humans, so we have 23 pairs of chromosomes, out of which one pair is sex chromosome, remaining 22 are autosomes. So all these 22 or 23 pairs, the segregation pattern of these, they are independent of each other. Then the second one is chromosomes from each homologous pairs, they are sorted randomly. So remember when we were discussing about Mendel's laws, then he's mentioned about law of independent assortment. So likewise, there's the chromosomes, the homologous pairs, the segregation pattern of the homologous pairs are independent and they're sorted randomly during the formation of gametes. Then the third point is that the parents, they synthesize gametes which contain only half of their actual number of chromosomes by meiosis. So gamete formation always involves meiosis, which is a reductional division where the chromosome number is reduced to half, and then during fertilization then the cell becomes, the fertilized cell or the zygote becomes diploid with one pair from each of the fusing gametes. So Morgan and his colleagues, they conducted experimental verification of these chromosomal theory, and the organism they selected was fruit fly. Morgan and colleagues, fruit fly, which is also known as Drosophila, Melanogaster. So Morgan selected Drosophila. There were certain reasons why Morgan selected Drosophila melanogaster as the experimental organism. The first one being, it's a simple organism which can be used efficiently for genetic studies because the features, they can easily be seen on even a low resolution microscopes. Then second is these fruit flies or Drosophila, they have a short lifespan, which they cover their entire life cycle in about 2 to 3 weeks. So it's easier to study larger number of generations in a shorter span of time. Then they can easily be grown on synthetic media in laboratories, so it's easy to monitor them. And plus, because I've already mentioned about their short life cycle. So these are some of the factors which led Morgan to study the inheritance pattern on these fruit flies. And the features, the contrasting features, these fruit flies, they also have very contrasting features which can easily be varied. Plus one of the other important points for the selection of this organism was because of the sex determination, where the male and the female flies can easily be distinguished. Now we'll look into the sex determination, the basis of sex determination in different organisms based on the study which was carried out by Henking in 1981, where he identified specific features or specific nuclear characters that were responsible for imparting a particular sex in different organisms. So during spermatogenesis, first he studied that during spermatogenesis there were certain characters, certain nuclear structures which were present in 50% of the sperms, while they were absent in the remaining 50% of the sperms. And whereas now, he named these structures as X body, and later these were named as X chromosomes. So first one is XX and XO type. So this type is present in grasshopper, where it contains 12 pairs. So this grasshopper contains 12 pairs of chromosomes, or you can say 24 chromosomes. So 12 pairs or 24 numbers, 12 into 2. So males only have 23 chromosomes, not 24. So means it has 11 pairs plus 1 is single. So that some sperms, whereas all eggs which are produced by females, so all eggs have one X chromosome. X chromosomes are present in all females, eggs of the females, which have autosomes. Along with the autosomes, they have X chromosomes which are present only in in females. Okay, so some sperms, so what happens, some sperms bear 50% of the 50% of the sperms actually bear X chromosomes, while the remaining, they do not possess this X chromosome. So during fertilization, so all eggs have this X, whereas sperms, so half of the sperms, they have X, the others, they do not have X. So during fertilization, during fertilization and fusion and formation of gametes, so when this X fuses with this X, it forms double X. Double X is characteristic feature of females because it has 24 chromosomes, 12 or 12 pairs. Whereas when this X, which is present in eggs, it fuses with this sperms which do not have this X body, which is 50%, then it forms XO, which is a characteristic of males, which have 23 chromosomes. So this is how the sex is determined for grasshoppers. So now we'll study about XX and XY type, which is present in mammals, in mammals and in insects. In insects and mammals, where we have XX and XY type of chromosomes, for which identify the sex of that particular organism. So there's both males and females, they have same number of chromosomes. Like in grasshopper, there were unequal number of chromosomes in males and females, whereas in insects and mammals, they have equal number of chromosomes. X type is larger, while the Y type is smaller chromosome. This is large, this is small. Okay, the X1 is large, the X chromosome is large, while the Y chromosome is small. Now the male chromosomes, so the females, they have AA plus XX, while males, they have autosomes plus XY. So here in these females, because they have XX, so we can also say that they are homogametic, means both the gametes, these are same, whereas females, these females, they are homogamic, whereas males, they are heterogametic in nature, because all the eggs which are produced by females, they will have the same X chromosomes. That is why they are homogamitic in nature. Whereas males, they are heterogametic. Why? Because some of the 50% of the sperms will have X chromosome, while the remaining 50% of the chromosomes will have Y chromosomes. So this is why this is heterogenetic in nature, heterogametic in nature actually. Then we come to the third type, which is ZZ and ZW type, which is present in birds. So here it's the reversible. Although here also both males and females, they have the same number of chromosomes. But unlike insects and mammals, where males are heterogametic and females are homogametic, in case of birds, the females are AA plus ZW, which is the females are heterogametic in nature, and males, they are AA plus ZZ, means males are homogametic in nature. So in case of insects and mammals, the sex is determined by the sperms, which will fuse with the eggs, whereas in case of birds, the sex is determined by the eggs, because the eggs are heterogametic in nature, and which egg fuses with what sperm, so that will determine the sex of the birds. So this is all about the chromosomal theory of inheritance, and then we'll discuss about about linkage and recombination and various genetic disorders in the upcoming video. So if you all have any doubts and queries, please come to me, please you can ask me in the comment section or on the email ID, and I'll be happy to answer all your queries and doubts. Please hit like, share among your friends if you find the content useful, and do subscribe to the channel for more such videos. Thank you.